Welcome back to our class. It's M.SAIDI. As usual, today we are going to be looking at the prediction of grade 11 paper. Please take note of this. This prediction will help you to get a distinction whether you are in Gauteng, Limpopo, Northwest, or any other province. Yes. So, and the days where you are writing your papers, they're different from one province to another. So please use this prediction so that you can obtain your distinction. In grade 11, paper 1, we have specific topics we are supposed to look at. Basically, we have photosynthesis, animal nutrition, respiration, gaseous exchange, and excretion. As I've been going to different schools to help out and then prepare different students so that they are ready to write their final exam for this grade 11. So I'm here for live so that I also help those have not gone to their schools. So if you look at this, there's something which is unique about these topics. Photosynthesis, nutrition, respiration, gaseous exchange, and excretion, all of them are processes. So it means that if you look at paper one is made up of what? Processes. Photosynthesis, so we're going to look at it. We has 32 marks. So it means that you have to put more effort there. And then this is also 32, 32, 32. But why this one is having fewer marks? So what are you supposed to focus on so that you can score the 32 marks in photosynthesis? So let's go to our distinction material. Guys, when you come to photosynthesis, you need to know the phases which are involved in photosynthesis. That is the light phase and the dark phase. But in most cases, if we don't bring these phases, we bring the factors which affect the rate of photosynthesis. So we like to bring about these experimental questions which are involving the factors which affect the rate of what? Photosynthesis. So these factors, with their experiments, how does the rate of photosynthesis affected with the increase in light intensity, the concentration of carbon dioxide, and then the temperature? These factors, these graphs, please take note of them. Sometimes we bring them when it is an experiment. So please don't go without knowing how these factors operate. You have to know how these factors operate. That's number one. Then number two, we bring, apart from the phases, bring about this diagram of the chloroplast. Please know how these chloroplasts work. Adaptations of the chloroplast to perform its function of photosynthesis, please don't go without knowing it. Yes. So sometimes we also ask you this. Even last time I talked about it for these people who are, yes, this, what is ATP in full? Sometimes we call it energy carrier. Yes. So please take note of that, it contains the energy. It's the one which break down to produce the energy which is needed in the reaction. So please go when you know about that. So the factors which affect the rate of photosynthesis is very important. Don't go without knowing them. Then number two, how do they operate? Like increase in this, what happens? So if they bring the experiment, because we like to bring the experiment, please know how it works. Yes. You know this experiment which we usually bring for light. They show you the light and then it is I think I'll just post some questions so that you can try to practice them so that it makes your life a little bit easier. Yes, so photosynthesis, the 32 marks. So number one is going to come from, or it comes from, apart from these terminologies. Number one, adaptations of the leaf to carry out its function, that is maximizing the absorption of sunlight, so that photosynthesis is taking place. That is the first point you need to know about. Then the second point, the structure of the chloroplast. Chloroplast, guys, is it has these pockets. Now, it's a little bit different from the mitochondrion, which looks like a hot dog. Yes, when they bring that structure, you have to know that that's a what? A mitochondrion, which has, it's like a hot dog. Yes, while the chloroplast, the major difference is the mitochondrion, it looks like a what? A hot dog. It has, you know, when the hot dog, you put the, how do they call it? Mustard. Yes, you put mustard, you cut the hot dog, you see, it looks like it. I'm not saying that this is the structure, but I'm just showing you to see that when you talk about the mitochondrion, if it is drawn and ask you to identify, please, you have to know how it is supposed to look like. And then a chloroplast, it has these pockets, you see them. Yes, it has those intergranular lamella, you see. So that's the major difference between the chloroplast and the mitochondrion. It's also double membrane. So please take note of that. Then we go to the next thing, light phase and independent phase. Yes, just know that light phase and independent phase, please take note of them and what happens in each. So that's not that's a much big deal. But when it comes to the difference between light phase and the dark phase, at least go when you know two of them differences. Yeah, if you are to ask too many, we don't ask more than three. Yeah, in most cases, that's how it is operated. Then this is very, very, very important. I repeat, the light phase and the dark, the factors which affect the rate of photosynthesis, how the rate at which photosynthesis is taking place. These factors, please, 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 don't make a mistake. If you go to the paper without knowing those paper, how do those factors, how do they operate? You are making a mistake. You are making the biggest mistake. Yes, please don't go without knowing that experiments of light intensity. This is the first experiment. This is the second, and the experiment we like, and then this experiment. This experiment, we like it as an investigative question. Yes, if it does not come, doesn't mean that in your province it will come in another province, because we like it. We like it. So yes, so please don't make a mistake to go without knowing how this experiment operates. How does it affect the rate of photosynthesis? I think I'm done with the photosynthesis. When you go to animal nutrition, what are you supposed to, where are you going to get these 32 marks? When you go to animal nutrition, yes, this is fine. Physical digestion, chemical digestion, you need to know, but the structure, this structure of how to label the parts, please make sure that you know how to do those parts. Yes, chemical digestion and types of digestion, whereby where does this mechanical digestion or physical digestion and the chemical digestion, where do they take place? Yes, and what is involved in there? Please make sure that you know it. Peristalsis, not that a big deal, but yes, we can bring it. There is this. Don't make a mistake without knowing this, the adaptations, this absorption process, whereby basically not absorption process, but adaptation of the villus for its function, very long, what, all these factors. Yes, please don't go to the paper without knowing the villus. Yes, it's very few papers where we sit and we don't bring this virus. Make a mistake without knowing the adaptations. How is this virus adapted to carry out its function? Then you're losing marks. Please don't go without knowing that. Then number two, the labeling of the digestive system. Yes, so the 32 marks are going to come from there. Most of them are going to come from there. And if the 5 marks are there, yes, it will come from the homeostasis. We like the glucose too much. How does control of this the negative feedback mechanism of glucose? Yes, don't make a mistake without knowing that. Please go when you know that. This don't expect that to come because it is next year now. So digestive system, when we talk about we say that the 32 marks, please go when you know how to label the structure of the digestive system. Please know how to do the villas, yes, how to label it, and then the adaptations of the villas, and then also chemical and physical digestion, then plus the negative feedback mechanism or the homeostasis of the glucose. Yeah, then you have your 60 marks. The paper is cheap. Paper one for case of grade 11 is the cheapest. Yes. Why? Because it involves labeling, drawing. Yes. So please, please, please go when you know that. Then let's go to the next topic, which is respiration. Respiration is a little bit fewer marks. Why? Because the content is not that big. Therefore, we expect to have fewer questions. However, we bring these questions. Yes, the sausage is here, or the hot dog. You see this. Please, please make sure that you know the structure of a chloroplast and how what is the function of the chloroplast. Yes, the function of chloroplast is produce energy, ATP, in energy in form of what? ATP. Please, please go when you know how to deal with the chloroplast. I'll do a recap at the end, and then if you don't have this booklet, download it and then use it. Né? Yes, you'll find a link there where it shows join. Yes, after that you pick on the books, then you'll be able to get the book. Yes, they are saying that the process of cellular respiration, this glycolysis, Krebs cycle, oxidative phosphorylation, you need to know them, but the way how we bring it, in most cases, I'm saying in most cases, we bring it as a combined, yes, a combined, like this, yeah, at least you know how it operates. Or we combine respiration with photosynthesis. Let me show you. Let me show you what I'm trying to say. We combine, yes, here is respiration, and then we do something like this. And remember that the product, if we are saying that photosynthesis, you have carbon dioxide plus H2O, giving you the CH6, H212O6 plus oxygen. This is the one which is being used in what? In respiration. So the product here is being used here. Therefore, I'll have CH6 plus H2O6 plus oxygen, giving me carbon dioxide plus H2O plus ATP. You see? And then now this is used here. That's why we like to interact them. Yes, we like to interact them. Please go when you know how this interacts. Yes. So respiration, yes, please, please, please go when you know how those two operate. What is aerobic respiration and anaerobic respiration? Yeah, those are simple definitions which you have to know before you go. Then we go to the next question, whereby they are talking about the next question, whereby they're talking about the next topic, whereby they're talking about gaseous exchange. Gaseous exchange is very short, precise. So basically easy to understand and easy to pass. So make sure that you maximize the marks there. There are no processes that are difficult in gaseous exchange. So make sure that you get the 32 marks from there so that you score a distinction. Yes. Photosynthesis, gaseous exchange, guys, you can do it. You can do it. We need to see you in grade 12 next year. Yeah. We need to see you in grade 12. Yes. Yeah, you can do this. You can do this. All right, let's continue to gaseous exchange. What do you need to look at gaseous exchange? Did I talk about the experiment for the case of respiration? Yes, this experiment, we like it, we love it. know how it operates. Know how it operates. If you don't know how, go to YouTube, type the topic you want, then you say ThunderEDUC, end with the ThunderEDUC. We'll bring all that information with this, trying to explain this. Please know that. Okay, let's go to gaseous exchange. Gaseous exchange, you need to know the factors which affect the requirements for effective gaseous exchange surface. Yes, this. Don't make a mistake without knowing this. Please, large surface area thing. Don't say large. Large for what? Large surface area for what? You have to tell us what it does. Thin and permeable. What is it for? So that the effusion can take place easily, because adaptation is a characteristic and its function. So it brings about the adaptation. So please don't make a mistake. Go to the paper without knowing the requirements for effective or efficient gaseous exchange surface. Yes. Then, yes, you can talk about the labeling. Yes, but. Yes, when it comes to this, please know this. Make a mistake. The structure of the alveolus. The structure of alveolus. Yes. Yes. Please, 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 don't go to the paper without knowing this, without knowing how this operates. Yes, you can even practice those questions so that you can score distinction. This is what I'm saying. This is what I'm saying. You have to know, yes, how is this alveolus adapted to its function? The adaptations of the alveolus for its function. It's the same thing as the the efficient gas exchange surface. It's the same thing. Please. Yes. So know how to label what is going in and what is going out. Yes, at least what is going in here, what is going out is carbon dioxide. Please know that. Yes. And then adaptations, please know that. Yes. So breathing in and breathing out, no one. If you talk about inhalation, is where there is contraction, there will be relaxation. Just know one, and then the other one will be the opposite. You don't need to cram everything. Yes, just know one. Yes. Then this I've talked about. There is this. What is this? This is exchange of substances in the tissue. Yes. So sometimes we bring this, and then it shows the whole thing, and then we hide. We hide this. We hide this. We hide this. We hide this. And then you have to know that, okay, this is inhalation, air in the lungs, oxygen goes to the blood, oxygen goes to the cells, carbon dioxide is pumped out, carbon dioxide in the lungs, and then is exhaled. So please, you have to know that. Yes. That alveolus, this adaptations. Yeah, here you know one. Yes, you have to know one. And then adaptations. Yes, this. Yeah. Don't make a mistake. Don't make a mistake. I'm not saying that don't read others. No. You must read others. But I'm talking about the things which, yeah. This one will come back in grade 12. And then if you make a mistake, you don't know in grade 12, it means that still you will fail it in grade 12. So you can do it. So this, sometimes it confuses the students, is also homeostasis of carbon dioxide, yeah, of breathing, or this one doesn't have the other side. Why? Because we don't need carbon dioxide in the body. Yes, that's why you don't say that when there's low carbon dioxide, when there's high carbon dioxide. No, when it's only high carbon dioxide, it's supposed to be expelled out of the body. Yes. So not that a big deal, but yes. Go when you know it, Yes. Excretion. This one is nice. It's nice. I've talked about a number of videos concerning about this. Please make sure that you know the following. Number one, Know how to label this. This is easy, né? Kidney. Yes, how to label this is also easy. So, when you talk about this, you can know how to label. Labeling is fine. This, know it, know it, know it, know it full. Why? Because we divide it into two. The male pigeon body. This, the male pigeon body. And how is it adapted to its function? This, I repeat, this. There is no way how you're going to skip a paper without knowing it. Make sure that you know it in detail. And then the formation of urine, we're not going to ask you all process. We have to ask you a few questions, né? Yes, so make sure that you know that. And then number two, this negative feedback mechanism or the homeostasis of water, salt, we can bring one, yes, yeah, but when you go back here, what are you supposed to look at very, very, very effective, effectively to get marks? Those marks, don't make a mistake there. Don't make a mistake here. Please make sure that you know this very well. Yes, if you are struggling, please go for these diagrams. These diagrams, you are going to get a distinction. This, yeah, make sure that you know it. You will be able to score a distinction. So you guys, you see that it is easy to get a distinction in this grade 11. Let me show you something. I wanted to, you see this, remember when you are bringing these questions, né? Some of these questions I've tried to discuss them. Make sure that you know it, né? Yes. And then make sure you know everything here. And make sure there's something which I want to show you. Make sure you know this. Yeah, I've tried to do a lot of questions concerning about this. Guys, go and find it on YouTube, né? ThunderEDUC excretion or questions about this, you'll find it. Just type at the end, right, ThunderEDUC. There is a adaptations of this for its function. Guys, don't make a mistake. You see, we like to ask this. Why is this large and this is narrow? This is a little bit wider and then this narrow. Why is it? Why do we have this coiled? Why is this in bow shape? It's bow shape for what? Why is this large? Why this is narrow? For what? You must know those things. The adaptations. Why is this coiled? Why is this coiled? Why is it coiled? So basically, please. And then why is also this coiled? This to convert tribute. Know how to label. Don't make a mistake, guys. We're not setting papers to kill you. We are setting papers so that you pass. So I think I've highlighted everything. Let me see if I have something which I've not talked about, and you'll find it there. Guys, these factors affecting, these requirements for effective gaseous exchange, please make sure that you know it. And then, this experiment, yes, make sure you know it, how it operates. Aerobic and anaerobic respiration, some the differences, at least you know 2 or 3. 2, they are good to go. Yes, don't memorize the hundreds and hundreds. No. Know how this operates, and then I showed you the other diagram, how they interact with each other. Yes, what is glycolysis? Where does it take place? And then cycle, where does it take place? Yes, oxidative phosphorylation, where does it take place? Don't make a mistake without knowing this. Yes, and what is its function? Yes. What is the importance of energy? You can also talk about it. No, 2 is good. This one is not coming. Then this one is the negative feedback mechanism of glucose. We say that the first one is, if this is high, the last one must be low. If this is increase, check blood glucose. No. The arrow goes like this. If the blood glucose, if it's drop, the last one must be increased. So here is you talk about the gland and then the function of that gland. That's it. Then it connects. The same thing here. Just highlighting you what you're supposed to remember. This, don't make mistake. Adaptations, don't make mistake. Yes, we talked about that. Yes, peristalsis is mechanical and chemical. Where do they take place? Know how to label this. Labeling the structure, the digestive system. Digestive system, know how to label it. Digestive system, don't make a mistake. You go without it. And then, yeah, sometimes we ask you, because this sometimes students, they tend to confuse them. Nah, which one is carnivore? This one is omnivore. Which one is herbivore? Also know them that herbivore, these are animals which eat plants only. Omnivore is plants and animals. Carnivore, these are plants. These they eat only meat. Yes. So please, and then when you look at its teeth, you can look, identify that this is this, and this is that. This experiment, we like it. I don't think you'll miss it out. I've been saying it weeks and weeks. Ne? Highlighting this in your classes. So now I'm just highlighting online so that those who were absent and those have not gone to their schools, at least you know exactly what you're supposed to look at. Yes. All right, let's go to the, yes, this experiment is also crucial, but we love the other one too much. Too much. I don't know why, but we love it. Yes. These factors, how do they operate? Please make sure you know it. Which one is the limiting factor when this one finishes? Then the other one automatically can't increase. So please know how those factors, light and dark phase, at least know two differences. Light phase, independent, light phase and dark phase. Know where each takes place at least. Yes, then this, have to know how to label. Know how to label, and then know the adaptations of it. Yes, to carry out its function. Then adaptation of a leaf for maximum absorption of sunlight. Sure, so guys, that's it. And then the remaining 2-3 minutes, let me just talk about the graph. Yeah, because when talk about the graph, yes, the first thing is we always ask it is line graph, line graph, line graph, bar graph, and then pie chart. Ne? So now, when you talk about pie chart, there is something which I need to talk about before I go to this, is percentage increase and percentage decrease. Guys, this, when they ask you percentage increase, is final minus initial over initial times 100. Then it's going to give you percentage increase or percentage decrease. Is it clear? Yes. If I started with the 20, I ended with 40, then I'm going to say 40 minus 20 divided by 20. Then I'm going to get 20. Is it 20? I hope so. I hope so. Which is 20 out of 40. Ne? Yeah, basically it's going to be 20 out of 40, which is 50%. So basically, is it 50%? 20 to 40. So this, if I subtract, it's going to be 20 out. It's 100%. So it's going to be 100%. That's how we do. But if now you started with the 40, you go to 20 out of so you end with it. So it's going to be 20 minus 40 out of, because I started with 40, then automatically it's going to be what? It's going to be a negative. So that means it's a percentage decrease. So don't stress if you see a negative. So that's it. The number 2, line graph. Guys, make sure that when you are plotting, it is nicely plotted. It's nicely plotted. Line graph, this is Y, this is X. Make sure that you put units. Make sure you put units. Caption must have the X factor and then the Y factor. Yes. We don't have a line graph which that. We don't have a line graph of that kind. If you find it, it means that you swapped the axis. Yes. Make sure that when you do the scale is maybe 0, 2, 4, 6. If the table is showing you 1, 3, 5, 6, don't start 1, 3. No. The difference is not the same. So just say 0, 2, 4, 6, or 0, 5, 10. It's not a must that you're supposed to start with a 0. Yes. 10, 20, 30, 40, depending on the lowest figure. Yes. So make sure that the difference is the same. Then if it is a bar graph, make sure that these bars are the same size, and the spaces between the bars are the same. Or otherwise, you're going to lose marks. Then if it's a pie chart, make sure that if they give you percentages, maybe 10%, 20%, we don't plot with the percentages. You convert this into 10% out of 10 out of 100 times 360, so that you use the protractor and then you measure. I did dedicated the full video concerning about this graphical work and how do you score marks in exam. So I'm just doing the highlight so that you remember. When you go to the experimental questions, investigative questions, independent variable, dependent variable, in most cases, when they bring the graph, the box, the first one in most cases is going to be independent. The next one is going to be dependent. So it means that ID, independent, dependent, it goes on x-axis, and then this one goes on the y-axis. But read the question nicely, you will get it right. You will never get it wrong. Yes. I think, yeah. Then how do you increase the reliability and validity? Please also go when you know that. Don't just say increase the sample size. Which size is it? Which size? Maybe they used 100 individuals, or they increased from 50 to 100, or how did they do it? They used. So if they ask you in past tense, you answer in past tense. If they ask you in present tense, you answer in present tense. Yes, guys, I wish you the best. This paper is gonna be easy. Make sure that you kill it so that you make me proud. Yes. Don't forget to subscribe, like, and share for our daily new videos. In grade 12, we will be live every single day so that we help you out. We help you out, and then you become a better, responsible citizen. This is M.SAIDI as usual. See you again in the prediction of paper 2. I think, let me first do, I'll come back on Monday for prediction, and then I'll come back for paper 2. Yes.